As I mentioned in the previous slide, each tool provides different set of Go terms, and only a few terms are in common with the experimentally validated terms. The next problem is that the experimentally validated set is incomplete due to the lack of knowledge or experiments. These problems have some consequences, like how would a biologist select a tool, or how would the developer test their tool. Metamorphic testing has been used to test programs in other areas which face the same or similar problems. In this study, we explored the feasibility of applying metamorphic testing to AFP tools. Metamorphic testing is a technique that instead of checking the individual outputs, it only checks the relations between different outputs. As an example, if the program under test is a sorting program that sorts a list of integer values, by executing the program with a source test case, we will get an output. And then by randomly permutating the source test case, we will have another test case which is called follow-up test case. And by feeding the follow-up test case into the program, we will get another output. At this stage, we will define valid relations between these two outputs, which in this case, in this specific example, the relation would be two equal sorted lists. So we expect two equal lists. If we get two equal lists, that would be a fail. That would be a pass, and otherwise that would be a fail. We refer to these relations as metamorphic relations. By applying metamorphic testing to AFP tools, we will execute the program with two source test case and follow-up test case, which can be sequences, and we will have two sets of Go terms. Then we check whether the metamorphic relation holds for these two sets of Go terms. As an example, if the source test case is the canonical sequence of protein tyrosinase, by executing the tool with this sequence, we will get a list of Go terms. And assuming that the follow-up test case is a disease variant of protein tyrosinase, we will get another list of Go terms. For this specific example, biological knowledge says that we should expect a change in the Go terms, in the output Go terms, from the source, the Go terms set 1 to Go terms set 2. So if we see a change, that would be a pass. Otherwise, that would be a fail. To evaluate the feasibility of applying metamorphic testing to AFP tools, we picked the top performing tools from CAFA 2. We started with 28 tools. At the time of investigation, only 3 tools were available or would work. So we contacted 25 authors and sent them a list of sequences and asked them to give us the output of their tools. 12 authors responded, and 6 out of 12 authors gave us their results. So we finished with 9 tools, which are mentioned here. But what did we send them? We sent them 18 sequences from 3 proteins. For the first protein, which is tyrosinase, we sent them the canonical sequence and 7 disease variants. For the second protein, which is cytokine receptor common subunit gamma, we send them the canonical sequence and 4 disease variants. Finally, for the third protein, which is toll-like receptor 4, we sent them the canonical sequence and 2 natural variants and 2 splice variants. For each of these, we consider the canonical sequences as source test cases and each of the variants as follow-up test cases. So in total, we will have 15 pairs of source and follow-up test cases. These are the results of running those 9 tools with 15 sequences, 15 source and follow-up test cases for the molecular function ontology. We see that 4 out of 9 tools fail all test cases. Blue shows passes and orange shows fails. And only 1 tool passes all test cases. For the biological process ontology, the same 4 tools fail all test cases again, and only 2 tools pass all test cases. So, in this study, we are exploring the feasibility of applying metamorphic testing to AFP tools, and we saw that it's possible to create metamorphic relations based on carefully selected protein examples. And we also observed that several tools did not pass all test cases. So we believe that AFP community need to work together for quality assurance. We also conclude that metamorphic testing seems to be a good way for testing AFP tools. For the future works, we will communicate the authors, and we will do a post-analysis to find the actual change from the source to the follow-up. And we will also explore more specific metamorphic relations, like the actual change, instead of just checking is there a change or not. We plan to develop an expanded test suite by exploring the feasibility of developing metamorphic relations for cellular component ontology, and by developing a test suite with a large number of protein examples, and also creating test cases for human phenotype ontology term prediction tools. We will also work with the AFP community to expand the number of tested tools. And eventually, we will develop a metamorphic testing framework which is readily available for the users and developers. At the end, I would like to thank National Science Foundation and the organizers of Function Cozy for the travel fellowship, and PIs and the teams developing EVEX, Orango FunFams, Sifter, Jones UCL, Pacano Lab, Profun, Panda, CBRG, Gorby, Fango, Cons, and PFP. And finally, the organizers of ISMB, Function Cozy again, and the reviewers for their valuable comments. This is our team, and we also have a poster number A-169, and I invite you to visit our poster, and if you have any questions, I would be happy to answer your questions. Thank you.
Questions?
I have a comment, actually. I thought this was fantastic. It highlights quite a few problems that I'll talk about. Sorry.
So I think this was great. This highlights quite a few problems that we are probably having as a community in CAFA. And, you know, one thing is probably we should do a lot more software sharing, or at very least results sharing. Of course, I'm going to ask you, what is the one program that performed perfectly, and you'll probably not answer that right now unless the authors are ready to. So it would be interesting to know.
Yes, not at this point, but
Yeah.
this evening that we have the poster session, if you are the PI or from the team developing the same tool, we can give you the results for just your tool.
Okay, so everybody heard that in the room, involved in these tools, so that's great. And yeah, I mean, I just think this is a great way of showing, of testing, and highlights the need for sharing software at the very least. Sometimes you can't because it's really, you know, just has been created and people are reluctant to share it for fault of cannot run it on any other platform really. But yeah, at the very least, try to share your results and operations. So thank you.
Thank you.